Okay, so it's going right now. Hit present. I'm going to go over your thing when you're ready.
All right, yeah. All right, so cytochrome BC and cytochrome oxidase, the two middle proteins in the ETS. So the ubiquinone transfers electrons 2 at a time to the cytochrome BC complex, which give energy for more hydrogen ions to go on the other side of the phospholipid bilayer through cytochrome BC. And then electrons go on the cytochrome C transport molecule to the cytochrome oxidase complex one at a time. And as soon as 4 electrons are inside the cytochrome oxidase complex, they take 8 hydrogen ions and 1 oxygen molecule, or O2, and create 2 oxygen molecules, and 4 hydrogen ions can then go on the other side of the phospholipid bilayer. And what that does is it further increases the hydrogen ion gradient that has been present, which James will talk about next.
Thank you, Frank. So I'm here to talk to you about the ATP synthase protein complex. This complex is contained in the thylakoid membrane and in the inner mitochondrial membrane. The FO unit, which is the top part of the protein complex, is contained within the membrane. The F1 unit is contained on the interior of the cell. This protein complex harnesses the hydrogen ion gradient that was created through the previous protein complexes in the electron transport system. So what this complex does is it harnesses the hydrogen ions by bringing in ions into this top FO unit, and each ion is contained within these little chambers that are shown here. Then, as a new ion is added in, the chamber spins and spits out one of the ions that was previously within it. So at all times, these all hold the protons that have been contained in the outer gradient. Then, as it spins, it actually creates the energy that is needed to create ATP through ADP and the phosphate group. So when three of the protons are transferred across the membrane, enough energy is created for the ATP to be synthesized. So this is constantly happening within the cell. These are embedded all over the cell, and there is constantly that gradient on the outside with the more hydrogen ions out here and the fewer hydrogen ions on the inside, and that is maintained through the previous protein complexes within the electron transport system.
This is a total list of all the reactants and products of the electron transport system. Going into the system are NADH, hydrogen ions, oxygen, ADP, and a phosphate group. Coming out are NAD+, water, and ATP.
So location. In animals and other such organisms, they are found in the inner mitochondrial membrane. And photosynthetic eukaryotes, they are found in the thylakoid membrane, things such as plants, as you can see here. And in bacteria, which are obviously the much simpler cellular types, they are found in the cell membrane, as you can see here.
All right, so the ETS was discovered and defined by David Keelan in 1925 in Moscow. And he defined it, or at least the ETS, by 3 hemoproteins. And he named these hemoproteins cytochromes A, B, and C. And these were later changed as we came to know more and more about the ETS process. And later, in fact, Kellen and his team of scientists in the 1920s and the 30s learned a lot more about not just the ETS, but cellular respiration and the respiratory chain as a whole. Yeah. So
Thank you for watching, class. We hope to see you next time.
Next time?
Yeah, probably. All right. That's good. We're done.